A very warm welcome to all the participants who have joined us for our 43rd session of BISMiS Live. We have an excellent speaker with us today, Professor Rup Lal, who is from University of Delhi, India. And he'll be speaking on tracing his lab's scientific evolution for the past 25 years from microbial diversity to genomics and beyond. Professor Rup Lal is an Indian National Science Academy senior scientist at the University of Delhi and a fellow of the FAAM, INSA, the fellow of National Academy of Sciences, the fellow of National Academy for Advancement of Sciences, and the FAMSC. He has served as the ASM ambassador for India from 2012 to 2015 and is currently the ISMI and the FEMS ambassador to India, as well as the ambassador to the IMILI South Asia Center. He began his academic career as a lecturer at Sri Venkateswara College, New Delhi in 1979 and joined the Department of Zoology at the University of Delhi in 1992. He has held key positions including the head of department, the warden and provost of the PG Men's and Mansarovar hostels. He was Dean of the Faculty of Sciences, Chairman of the Board of Research Studies, and also the Dean of Examinations, and he retired in 2018. He's been a visiting scientist at renowned institutions such as the University of Cambridge, EPFL, ETH, Zurich, University of Bielefeld. He's also received prestigious fellowships, including the Alexander von Humboldt Fellowship, Indo-US ASM Professorship in Microbiology, and the Australian Government Endeavours Executive Fellowship from 2018 to 19. His awards include the ASM Mozilio Carter Distinguished Service Award, AMI Lifetime Achievement Award, Best ISMI Ambassador Award in 2022, and the INSA Distinguished Lecture Fellowship in 2024. Professor Rup Lal has served as the editor-in-chief of the Indian Journal of Microbiology for over a decade, from 2006 to 2013, and is also on the editorial board of M Systems, ISMI Communications, Environmental Microbiology, ASM Spectrum, and others. He has contributed to the national and international committees, including CSIR, DBT, DST, and the Ministry of Environment and Forest and Climate Change, and was also on the review committee for the ASM IUSSTF, the Indo-US Professorship in Microbiology. He's also the former president of the Association of Microbiologists of India, and he currently leads the Indian Network for Soil Contamination Research. And he's worked extensively in microbial diversity, systematics, and ecology. So with this brief introduction, I have the privilege of inviting Professor Rup Lal. Sir, you can start your talk.
All right. So at the outset, I would like to thank Ben, Julie, and Aaron, and also Kamlesh for inviting me to this wonderful initiative. In fact, as I could see, actually, the list of previous speakers, they were which you all have selected. Actually, probably they were wonderful microbiologists and taxonomists. But as I mentioned with Kamlesh, actually, we got into this particular discipline not really that we wanted to do taxonomy, but it was just accidental that we started doing it. But now we are enjoying doing taxonomy, systematics, genomics, and beyond, which really have become really very important as of today. As I could see, actually, that this initiative by BISMiS is really meant for young students and also young faculty who really want to, BISMiS want to motivate them actually to get into this very important discipline and microorganisms, studying their diversity by using genomics and metagenomic approaches has really become very important. And as of today, as I can see, and I have been telling my younger colleagues that you cannot do good biology without understanding the role of microorganisms. And if you really want to study the role of microorganisms, you really need to study the microbial diversity by using computational biology. So probably I will go through the work that we had been doing just to let you know that it's not really that difficult to get into this particular discipline. Even microbiologists or biologists who are not really trained actually microbiologists in taxonomy or in computational biology can get into this particular discipline, and probably this is the right time that you really need to initiate actually getting into microbial diversity. As you all know, actually, this microbial diversity has really become very important because of very rapid evolution in DNA sequencing and also in computational biology. I will not get into details, but want to tell you that way back in 1988 or 1980, when we wanted to do the sequencing, it was so difficult for us that even to just sequence a single gene, we used to spend months and years together. But things have become so good now that everything is cheaper in terms of sequencing, and there's a very rapid evolution. At the same time, doing computational biology using tools that has also become much more easy. And as a result of that, just to ignite you and let you know what I said, that microbiology is really very important, and many new things which or microbes which were there with us, probably we did not know that their impact is so high and their number is so high that they really rule the planet. Now, if you look into the newer data that has come in, probably the total number of this organism, it will always fluctuate. Actually, on this particular planet is something around 0.9 to 3.2 into 10 to the power 30, which includes archaea, bacteria, viruses, fungi, protozoans, and also algae. And the number of viruses is something around 1 into 10 to the power 31 viruses on this particular planet. And if you look into the total biomass, rough estimates are that the microbial living biomass on this particular planet, microbes represent something around 30% or 27 to 33%. So these are the newer figures actually, which you can see the plants coming at the top, which are around 80%, and microbes really 16 to 30%. Now, even if they are at number 2, we believe that they are being at number 2, and because of they having very unique features, they should be considered as at number 1. So this is, as I mentioned right in the beginning, that microbes are really very important. Doing microbial diversity and looking into it, actually, the interaction of microbes with almost everything has really become very important. But most of the microbiologists or biologists, even today, probably they are reluctant to get into this particular discipline. So I wanted to just go through how did we start actually into microbiology. We had not been doing hardcore microbiology, but looking into the production of antibiotics in one of the actinobacterium, which we call it as Amicola typhus mediterraneae. But when we got stuck up, actually, we thought of getting into doing microbial taxonomy. So as you can see, actually, our first publication in ISG, JM, actually, it appears sometime in 2005, actually, when we did the first taxonomy of a bacterium, which we call it as Pingobium indicum B98. And subsequently, it became very fascinating for us. So every student actually, total 70 students who passed out, graduated from my lab, everyone has really started with doing taxonomy and systematics, looking into 1 or 2 bacterial strains and then publishing. So in all, probably in few years' time, that time, we could publish nearly 52 novel species. But when you looked into it, what exactly we are doing by using the traditional polyphasic approach, we realized that with this particular background, why not to get into genomics? So we started sequencing, became very cheaper, and we started sequencing the genomes, draft genomes, and then also complete genomes. But by doing that, we realized that now is the time, because of the availability of newer computational tools, probably you can get into comparative genomics. While doing comparative genomics, we realized that same tools which are being used to do the genomics actually can be extended to do metagenomics and also study microbiomes and systematics and so on and so forth. So even still, actually, we continue contributing to Burgess Manual or to taxonomy by using newer tools which are being streamlined or invented year after year. So that's what actually I just wanted to convey with these two slides, that it's really not that difficult. Of course, if you can do high-end computational biology, then there's no match for the type of work that you can produce. But even if you are not actually computational biologist, to start at this particular young age, you can really do number of things. So when you started doing computational biology, taxonomy, sometime in 2005, a simple approach, which you call it as a polyphasic approach for bacterial classification, was available for us. And we continued using this approach. But after some time, or in 2018, actually, there was a revision because of the limitations, and then a newer set of rules came in, and we started bringing in genomics. And this particular, there is a transformation in the taxonomy and systematic methods, and you'll find, as I will discuss later, that now actually we are including genomics, plus we are including in the protocol actually not only complete genomes of the type strains, but also of the genomes that have been published. Now, this polyphasic approach, it was, when we look back, we find that it was really cumbersome. Doing 16S, then going for DNA hybridization, and then eventually with the change in the methodology and bringing genomics into this, actually probably many strains which was classified by using this particular simple approach, they were reclassified. So there was really a need actually to shift from genome-based, 16S RNA gene sequence-based taxonomy methods to genome-based methods. And as we were discussing, actually, in India, probably we have a disadvantage in the sense that our National Biodiversity Authority is really very strict and enforcing very strict regulations. We are not able to really come out of it and exchange microbial material in order to do better taxonomy. But with the coming up of this genomics, and even including metagenomics into it, probably it has become much easier actually to get into microbial diversity. So just to tell you again, it was very easy actually for us to do taxonomy of bacteria. Just every student picking up 2 or 3 bacterial strains and looking into it by using DNA profiling, and then eventually, if they were able to find out that really it's a new species, then we used to go for DNA hybridization. And if it turned out to be less than this, what we normally did was publish the paper in a IJSCME. So that's how we did the first paper, which is published in the year 2005. You can see over here, we had 3 bacterial strains. One was isolated in Japan, the other one from India, the third one from France. And all of them were named as Spingomonas possimabilis. But they had a common character, that they had genes that were responsible for the degradation of HCH or lindane, a very persistent pesticide. So we were working with all these 3 genomes, and at one point of time, we decided to look into it, how these catabolic genes, they got into these particular 3 strains which have been isolated from India and from Japan and also from France. So when we started looking into using the polyphasic approach, I will not get into the details, we found out that they really are not very close to Spingomonas possimabilis, and their closer relationship is with Spingobium chlorophenicum and also with the Spingobium chucubicensate. So we reclassified them and named them as Spingobium indicum, japonicum, and francensis. So that was our first paper. And with that, we started doing actually, student, they could easily learn how to do the sequencing, how to assemble a complete 16S ribosomal RNA gene sequence, and then probably do DNA hybridization. And everyone really started getting familiar with these 3 or 4 molecular techniques. And with that, actually, we actually, in course of time, there was a need actually that now, because polyphasic approach has got a lot more limitations, so why not to use genomes into the, so you can see the revolution of methods or rewriting taxonomy, extensively used polyphasic approach, and then brought in actually now in 2018 actually the new rules have been set and they are in place where we are using the genomes, either draft genomes or complete genomes, into looking into the taxonomy of a bacterial strain. And now in 2005, I will discuss with particular examples that not only the type strains, but also the bacterial sequences, even from the metagenomes, they can also be included. Now, in 2018, which you all are familiar with, that the rules are really set in place actually to use genomic. I'll not get into the details, but use genomic complete genome or partial genomes in order to resolve the taxonomy of Amycolatopsis, which we had been a collection of such strains in our lab. And when we did actually by using 16S ribosomal RNA gene sequence approaches and also the genomic approach, we found out that Amycolatopsis methonilica, then also Erythrea and Thermophileba, they all fall into one group and were very closely related. So later, by using actually this particular genomic sequences also, we reclassified and named Amycolatopsis erythrea as a heterotypic synonym of Amycolatopsis thermophileva. So the advantage of using all these tools is that you really don't need to do sequencing yourself. There are a lot more DNA sequences that are already available in the databases, and you can revisit actually the classification. Similarly, we did the same by using same approach with Cyclobacter. We found out that there are 3 groups of these bacteria, new species, which are but named different. And by looking into 16S ribosomal RNA and also genomic sequences and also ANI and AI approaches, we could again revisit and reclassify this particular group. So that's what we had been doing. And with the coming up of a revised genome-based approach in 2004, we started using all that. I will not mention, probably already you are aware of the package actually that has been mentioned actually in order to do the reclassification. And one of the bacterium where we have resolved the controversy is Williamsia actually, which we published again in IJSEM. And also recently, we had been contributing to Burgess Manual of Systematic and Archaea and Bacteria. And we reclassified actually because of the year after year, you'll find newer genomes becoming available. So there was although Cyclobacter controversy, we resolved 2 years back. But again, when we started writing a chapter to Burgess Manual, we found out that new genome sequences, they have come in, and we have to revisit and use all these newer methods in order to resolve this particular taxonomical ambiguity. And this particular manuscript, Whitman invited us, and we are revising and have already submitted the revised version to Burgess Manual. So with all that background, simple background in the doing taxonomy, using 16S ribosomal RNA gene sequences, and then moving to genomics, we could easily become familiar actually how to assemble the genomes, draft genomes and complete genomes, and how to do the comparative genomics, not only taxonomy. So we actually sequenced around 30 to 40 genomes, where we found out actually that with the coming up of PacBio and new sequencing platforms, even we can get a complete sequence of the chromosome of the organisms, and also you can find out how many plasmids are there. And as a result of that, we could make out very interesting depiction, the presence of catabolic genes on the plasmids and their association with IS elements and so on and so forth. Even from Manikan hot spring, actually, we isolated thermus from where actually you did this complete sequence and assembled actually the plasmid and also the complete genome. Interestingly, we found out that the heat tolerant genes, they are present on the plasmid, and this particular bacterium we are further exploiting actually because the top DNA polymerase sequence that we got appears to be novel, and it has better activity as compared to the traditional. So not only that you can do diversity, you can do the taxonomy, but you can also, by doing comparative genomics, by doing genomics, you can look into newer features in the organisms which can be commercially exploited in course of time. And also, would like to mention and tell my younger colleagues actually that if you don't have money or you don't have funds, you don't really need to do the sequencing. You don't really need to buy the sequencer. There are a lot more information that is available in the database. There are a lot more sequences or genomic sequences that are being added up to the database. You can simply develop a concept, download the sequences, and if you are good in computational biology, you can really come up with very good results and publish good papers, and also come up with the newer genes that can be eventually exploited for a variety of purposes. I'll just give you one example, like what we did in around 2018, actually, when we didn't have money in the lab. We simply could design a concept that let's have novel Spingobium genomes from the database. We actually wanted novel Spingobium genomes from the rhizosphere, then contaminated soil and fresh water and marine environment. And using all computational, the package of tools which I have mentioned earlier, we could come up actually what are the newer or flexible genome or genes that have got into novo Spingobium, which is from marine environment, or the one which is from the ribosphere, or one which is from. So just by using those signature sequences, you can make prediction that this particular organism probably will, or sequence, it belongs to rhizosphere, or it has been isolated from the contaminated soil. So that's just what I wanted to convey here for younger people. If you are good in computational biology, probably you don't need to spend much more money in order to do sequencing. You can actually download the sequences and make a concept and start working on it. So that's what we did, published number of comparative genome papers on comparative genomics, and we still continuing this particular work. With that background, we realized that doing comparative genomics, the same actually computational tools in a bigger scale actually can be used to do the analysis of metagenomes or microbiomes. So these 2-3 slides, what I'm showing actually, the flow diagram, I will not get into details, but just I will tell you that yes, basically the same tools are being used to do the genomic analysis. A broader version of these tools then is being used to do the metagenomic analysis or microbiome. So all that probably once you are, we started from 16S ribosomal rDNA sequences, and then went to draft genomes, then to complete genomes, comparative genomics, and then metagenomics. So one of the metagenome which we did from chemically polluted site, where there were almost 80 to 90% of the chemical, no soil. We probably were under the impression that probably there will be no microorganisms. But when we did the metagenome of that particular soil, we found out that it's really a very dynamic contaminated soil where a lot more organisms like spingomonads, novospingobium, then pseudomonas, which have got the catabolic genes, they got enriched in that particular environment. And our functional aspect of the analysis revealed that yes, this particular soil contained, or the organisms that were found out in that particular contaminated soil, they contain plasmids, they contain phages, they contain IS elements, they also contain. So under a particular pressure of nearly 10 years time, the entire microbial diversity got shifted actually in order to start eating that particular chemical which was put by the industry into that particular soil. Not only this, one of my students who were really very good actually, he's in US and doing great work, Sangwan. He actually mapped the genomes with the metagenomic data and developed a new concept of developing core genomes and predicting how these horizontally actually the flexible or catabolic genes or other genes they are getting into the organisms depending upon what is the type of stress that is existing over there. Would like to mention and advise all young students when you are doing genomics and metagenomics and microbiome studies, please be careful that the selection of the organism is very important, like highly contaminated site or human microbiome or microbiome from hot springs, because then really you come up with very interesting results and you can publish a very unique and interesting paper. So we also got into metagenomics of hot springs at Manikaran, which is located at Himachal Pradesh at an altitude of something around 1700 meter, and the temperature is around 1936 degrees centigrade. We isolated a thermos actually from that particular environment. Not only that, we also isolated, took sediment and water samples and also did the physiochemical analysis. And when you looked into the diversity of this particular hot spring, we found out that there is an enrichment of Enterobacter and Bordetella. Now, when you looked into it, what is this Bordetella? We found out that it's a bacterium, gram-negative bacterium, which feeds on our gram-negative bacterium. And it was highly enriched. So from the metagenome, we could assemble the genome of this Bordetella vibrio. And when we assembled the genome of this Bordetella vibrio, we compared it with all existing sequences of this particular bacterium and found out that in the genome, one of the hit locus that was absent in this particular bacterium. And that hit locus is responsible, in fact, for the penetration of this Bordetella on gram-negative bacteria, on which it feeds. And when we looked into the sequence that we assembled from the metagenome, we found out that really this particular hit locus was missing. And when we did actually the wet lab experiment, scanning electron microscope, we found out that yes, the Bordetella vibrio that we have isolated, it really doesn't actually penetrate E. coli, but it simply sits on it. So our in silico studies were confirmed actually when we looked into the scanning electron microscopes of enriching Bordetella vibrio and adding E. coli to this. Not only this, as I mentioned in the previous slides, that these hot springs are also very dynamic environment where there were so many viruses actually that were also enriched. And when we did in silico studies, assemble the viruses, we found out that we could really assemble 65 phages and also relate them to which type of bacterial community those will be belonging. And when we did the enrichment and scanning electron microscope, you can see actually the phages sitting on bacteria. We didn't stop here. We went to around 300 meter up to Kir Ganga in the foothill of Himalayas in Himachal Pradesh, and also did the sampling over there, and analysis of the data revealed actually that yes, it is also a very dynamic environment, and probably a lot more biotechnological technological applications they can come up from this type of data. We are actually planning to do it with the industry, but our National Biodiversity Authority is not permitting us, saying that we do not actually permit microorganisms to be exploited even from protected areas. So that is the difficulty what we had discussed in the beginning, but that doesn't mean we should stop working. Even we did human microbiome by using same tools what I'm talking about and what I had mentioned in a couple of my slides, which I did not explain actually in detail. You can use to do the microbiome studies. So when you designed a concept, we selected actually tuberculosis patients, the normal, then those who were suffering from tuberculosis, and also those who were given antibiotics. So all the DNA were sequenced, and when we found out that the tuberculosis patients, there is a increase in the number of microbial community, especially actually you can see here Caprococcus and Bacillibacterium, which produce actually the short chain fatty acids and also propionate producers, and all these chemicals, they are responsible for impairing the immunity in the individual and also altering the appetite of the patient. So with this, actually, I just wanted to conclude and say that doing microbial diversity is really very important. Even those who are young scientists doing biology cannot do good biology without understanding the role of microorganisms. And if you want to do good science, get into this particular discipline. And BISMiS is really doing a great job, actually, by exposing you to the techniques and tools that are being used in order to do microbial diversity, microbial genomics. And also the other advantage is that if you handle the simpler system like microorganisms, you can easily move to complicated system. And no organism, no plant or animal is free from microorganisms, as you all know about it. I need not to mention. So with this, I thank you, Kamlesh, and also Juan and Aaron, for giving me this opportunity to just ignite young students that this is a very important discipline and you can really get into it. Thank you very much.
Thank you, Professor Rup Lal. This was really exciting. It actually shows very concisely how many areas one can work with if you work with diversity and systematics and metagenomics. It's really commendable, the hard work that you have put in and the number of fields that you have touched in this, presentation of yours. I wanted to actually, draw your attention to one of the slides that you just showed very briefly, where you said not enough criticism from colleagues.
That's what I didn't explain, Kamlesh. We were working on a very difficult problem where we wanted to produce antibiotic analogs and got a lot of funds and nothing was coming out and we were not able to publish.
Mm-hmm.
So that time we decided why not to do taxonomy.
Okay.
That's why I said accidental. But when we realized that doing taxonomy makes students to learn the basic techniques. They do sequencing, they do assembly, small, and then they do DNA hybridization. And so it's not really taxonomy, but and every student who used to come to my lab, first we used to give him 2 or 3 bacterial strains, and we also knew trick how to publish it, which strain we need to pick up to publish in a IGSEM, right?
Correct.
When you started publishing, almost some 10, 15 papers every year, then colleagues, they said, these fellows, they have now started industry of this publication actually and publishing. So there was a criticism that taxonomy is very easy, and that compelled us to move on to genomics.
Okay.
Your colleagues also, that criticism helps you actually to get into that. Made us realize that okay, let's stop it now, get into genomics.
Okay.
So that was a criticism from colleagues when we were publishing 20-24 papers in almost every year.
Yeah. So, my question was very specific to the last point that you made. As a microbial systematics, rather as a microbial ecologist, when you started and then you moved to microbial systematics, and when you started hearing this criticism that taxonomy is not a science or taxonomy is very easy, what would you like to say to those individuals who believe that microbial systematics is not a science or is not research? There is a lot of, rude criticism that we hear.
Kamlesh, I will not mention, but very prestigious institute, they started saying that taxonomy is no science. And that was very painful.
Right.
Because that's why I just wanted to tell my young colleagues that just by starting doing taxonomy does not mean just to do now. There is a revolution actually. Then there is a evolution of the whole approach now. Taxonomy is linked to genomics, to comparative genomics, to metagenomics, and microbiome. So there is no end to it now.
Yeah.
Probably to those, this is a probably now many people who were criticizing the taxonomy in United States, they are doing taxonomy and they are doing microbiology, even those who were geologists and plant biologists, because they cannot do good science without understanding the role of microorganisms.
Yeah, yeah. So, when you switched over from doing ecology and then you moved to classical systematics using polyphasic approach,
Yeah.
how was the transition taken by the lab actually, by the students who were in the lab at that time?
No, actually, when we were only sticking to one problem or two problems which were very difficult. Cloning that time, genes, either you are able to clone or you are not able to clone, right?
Right.
If you are able to clone, then you are not able to express it. So
Mm-hmm.
that used to be a work of years together, and many students after 3 or 4 years or 5 years having worked, very brilliant student worked in my lab,
Mm-hmm.
they used to criticize me that they came with a lot more ambitions and even they are not able to publish a single paper.
Okay.
That's why I decided that, okay, let's get into some more and editors of IJSME, and they were very helpful guiding us that you have done mistakes that time when we were not really familiar with taxonomy.
Mm-hmm.
So that's how students also, although they did their graduation or PhD, were very happy once they were out actually that they had 3, 4 publications and they were convinced that they learned a couple of techniques by doing this.
Right. Right. Okay. Um, so we have received a question that I will take. Um, the question is from Asif. I would like to thank you for such a wonderful presentation. I appreciate Professor Rup Lal's efforts to come up with interesting facts about bacteria. Asif wants to know about bacteria eating E. coli. How a bacterium can cannibalize its own type of species as nutrition. Is it possible to manipulate for disease management? Example in plants or human disease.
Actually, my focus was only on taxonomy and not because over the time when you work, you have so many slides you don't want to leave. So my slides were also in hundreds, so I didn't want actually to take much of your time. Now this Bordello vibrio, which I mentioned that it simply penetrates the gram-negative bacterium and there it multiplies. It's like phage is entering into a bacterium and then multiplying, and bacteria simply burst and dies. So same way, Bordello vibrio is a gram-negative bacterium. It feeds on all gram-negative bacteria, and people are working on it to use it as a live antibiotics or even to kill pathogenic bacteria in the sewage. So that aspect I did not really mention because this itself would have been a complete one lecture. But yes, it is being used as a live antibiotic and also
Okay.
being developed as a bacterium that can eliminate gram-negative bacteria, probably pathogens.
Okay. Okay. Fanas has a question. Fanas, you can take.
Thank you. Thank you very much for your talk. We really enjoyed that. I've got one question and then a comment. The first question is how do the regulations in India of sending out cultures and things impact on your normal taxonomic work? This is something that we have in South Africa as well, that we cannot just simply submit the cultures to different culture collections. And my question is, how has that impacted on your work?
I know when we published a couple of papers, that time probably it was not, the NBA rules
Not
were
yet.
not in place so strictly. So we published a couple of papers that time. But now, Kamlesh, and we were discussing before it started actually this particular lecture, that our Indian National Biodiversity Authority, still they are very strict, but they have come down to the level that in case you want to send a microbial culture outside to the culture collection, then please don't intimate us. So what we normally now do, we intimate them, they never reply, and we consider that as a positive reply and publish, but that is also very risky. We don't really do that because we don't get any positive reply. So still this debate and controversy is going on, and people who are dealing with taxonomy or even those who are in IGSCM, they were not very happy with the Indian approach, actually, not to share this bacteria in order to do taxonomy, but that's very unfortunate. But to overcome that problem, probably for younger colleagues, what I said, you don't really actually need to isolate and do taxonomy. Why not to revisit the taxonomy? Because there is after every 10 years or 5 years, the rules are computational tools and sequencing becoming so cheaper that you have a lot more data that you can revisit and reclassify the organisms. So that is one thing which can be easily done without getting into any controversy.
Yes, thank you very much for that. I think, India, South Africa, Brazil, and a number of other countries have that same problem. And that's why I want to make the comment, moving to microbiomes has been very great, because then we get also sometimes isolates and full genomes that we can use. And I mean, I've just seen this week that the GDTB updated, and they now have estimated 144,000 bacterial species, of
Yes.
which only, say, probably 25 to 30,000 has been named. So there are quite a diversity out there, and people can do a lot of taxonomy. And I think it's important. I am, as a taxonomist, feel that you need to give these things names, because the names serve as the anchor for the publications and things. And that's why I, as a South African, start using the C code, where I use bacterial genomes from an isolate, but using the C code to just name them, because that's the way that I can do it. But I think, with this metagenome data, we can actually do a lot, where you can do ecology. So your species description is not divorced from what's really happening. And I think that's where the great future lies, is being able to bind ecology, as you've shown, ecology with taxonomy.
Yeah,
So
but at the
thank
same
you very
time
much.
now this has become much easier, because earlier to do DNA hybridization in the polyphasic approach, we certainly required actually type strains from different culture collections, which was very cumbersome. But now digital DNA hybridization, with this coming in, probably things have become much, I think you will agree that much better now.
Yeah, and doing ANIs is, for me, quite an important thing. And I mean, we do it a lot, and it makes life so much simpler, because you can easily compare with a range of species
Right.
that you've never would have necessarily got hold of in your lab. So thank you for this.
Thank you so much. I guess I just have one question, Professor Rup Lal. When we work with metagenomes, we obviously do not have the isolate at hand. What, in your opinion, is the best approach to study the physiology based on just the genome data?
Right. Kamlesh, of course, every system will have plus and minus points, but there are now companies already which are simply sequencing from different niches and keeping the sequence with them. Now you just ask them actually that we need this type of gene for commercialization. You need to pay, and they give you the complete access to the sequence where they feel, yes, this type of gene could be there. Of course, in totality, when you want to actually study the physiology, when you do it with a single organism that has its own advantages, you will certainly miss a couple of things. But then there is also an advantage where you are getting a gene pool to be exploited for a variety of things. Probably wait for 5 to 6 years. You will have so many things available in terms of genes which you can simply buy and then express it. And if you want to produce an antibiotic, that will happen. Believe it, Kamlesh.
Yeah, that's true. I mean, the way the technology is advancing, genome
Yeah,
editing is
yeah,
becoming much more
yeah,
easier.
yeah. And
Copy and
all,
paste, pasting
even
right
for
somewhere.
human health, and probably you will realize that it will be more on microbiome based, actually. Many treatments. Yeah.
Right. Thank you. I guess there are no more questions, sir. Thank you so much once again for this wonderful presentation and an insightful talk that actually helped students understand the variation in the work that one can do just with diversity, metagenomics, systematics, and taxonomy. So thank you so much, and I hope you have a wonderful day ahead. And looking forward to seeing you at the 2025 meeting in China, sir.
Sure, it will be my pleasure. Thank you very much.
Thank you. Thank you so much.
Thank
Thank
you.
you, everybody.